The chapter 16 one is due next. I think it's not due till next Friday. I didn't look this morning. Has anyone taken the chapter 16 immune disorders quiz? Like, no, it's not till next week. Why would we have taken it? All right, so last class we got through about half of the PowerPoint, looking at different types of vaccines, how they're different, how they work different. And the second part of this PowerPoint, which we just barely touched on, was using that immune system response, the fact that we can make antibodies against antigens that we've seen to now test patients. So all we got through last class was defining these terms, the fact that we can test specifically for an antigen present in a patient, or we can test to see if a patient has antibodies. And we can use this to diagnose a patient. We can say, yes, you have the COVID antigen in your body, you actively have COVID. Or, oh, you have a COVID antibody in your body, it means at some point you were exposed to COVID. Again, indirect testing that's testing for antibodies gives us a lot of information. It's always good to know what patients have been exposed to, meaning they now have an immune response against things and in theory are immune to certain things for a while, but it doesn't always tell us exactly what's going on at the exact minute. So it kind of gives us a nice picture of overall health and where immunity is on things, but just not, what's going on at that exact moment. Now, some different types of tests. Ignore the page number. That wasn't with the ebook page number. The first type of test is called a precipitation test. Now, it's called this for a reason. When you think of precipitation, what do you think of? Rain. Rain, snow, sleet, everything we probably get in October. I think we've all had all of this already in October. It was sleeting the other day when it was like snowy and sleeting. Whatever. Precipitation, the word itself just means to like fall from. So any kind of precipitation is anything that falls from the sky. So the idea is that we are going to take insoluble antigens in a sample. So these are very small things. Not insoluble, soluble antigens, so things that are dissolved in a patient's sample. We're going to mix them with some antibodies. And if the right proportion of antibodies mixes with the antigen, it will fall out of solution and it will sink to the bottom, which is why we call it a precipitate. It fell out of solution, just like precipitation means it falls out of the sky. Now, there is a balance of the number of antibodies to the number of antigens that creates that biggest precipitate. And reason for it, if you only have a couple antigens, these little green circles up here, but you have a massive amount of antibodies, like the ratio is like 5 antibodies for every 1 antigen, that one antigen is going to get completely coated in antibodies, and it itself, although it is bigger and may start to slowly fall out of solution because it's now bigger and heavier, it's still not a very big molecule. And on the opposite side, if you have way more antigens compared to antibodies, again, probably like 5 to 1 antigens compared to antibodies, one antibody can hold on to 2 things, and it will do that. And it may still somewhat fall out of solution because it is now bigger, but it's still not that big. But if you get the right proportion of antibodies to antigens, those antibodies are going to attach to 2 antigens, and other antibodies will also attach to some of those same antigens, and they form these big, huge, large structures. They're larger, they're more likely to fall out of solution. So this tells us a couple things by doing this. One, by seeing anything fall out of solution, we know that that patient has that particular antigen. By controlling how many antibodies are added to the solution and looking to see where does it fall out of solution, we can also start to quantify as well. So we know that there is a ratio where it starts to fall out of solution the most, and we can play around with those ratios to figure out where antigen levels are, and we can even flip it and where antibody levels are at. Now, there's another similar idea to precipitation known as agglutination. They both have the same basic idea of what's happening. The big difference is what are they pulling out of solution. So precipitation, it is clumping of soluble molecules. So like little tiny things. Things that can be dissolved in a solution. They're soluble. Agglutination is clumping of insoluble molecules, so larger things. They are the same basic idea. It's just precipitations, small little soluble, agglutination, larger insoluble molecules. So in this picture, the precipitation is clumping, this big, huge structure with these little tiny soluble molecules. Agglutination, bacteria are not soluble. They don't dissolve in a solution, but we can still clump them together, and it makes it really easy to even visually see the clumping happening. So this is actually showing up on the very top. It was we added some antibodies to a patient sample, and the antibodies were specific for a staph bacteria. And because that patient had staph bacteria present, we saw clumping super easily. And we're like, well, the patient has staph. If we don't see any clumping, no staph. And so we can easily visually see this. So agglutination is the cross-linking of antibodies with just solid things. Super easy to see without doing anything else. And one common place we use agglutination, that idea of using antibodies to clump these insoluble large things together, is in something known as hemagglutination. And hemagglutination is we're clumping of red blood cells. Now, you guys take an advanced A&P? Some of you are who's in advanced A&P right now? I'm so sorry. For people that did take advanced A&P, did you guys do blood typing? Or have you done blood typing before, where you prick your finger and mix it with different solutions? So I see in advanced A&P they do it. I think in general bio they do it as well. The idea is that if you take a drop of blood and you mix it with different antibodies, your red blood cells have antigens on them. And if you mix it with antibodies, those antibodies are going to clump your red blood cells. So this is just showing what the drop of blood looks like on the little card. If we mix that drop of blood with antibody A, clumps, because antigen A was present. If we mix it with antibody B, there was no antigen present for B, so no clumping, doesn't look like that. And if we mixed it with antibody D, which is also known as the Rh factor, it clumps there as well. What is this person's blood type? Positive. Because they have the A antigen, it clumped with antibody A. Because they had that Rh antigen, also known as the antigen D, it clumped there as well. That's where we get positive or negative. You either have it or you don't. So we use this to determine blood type. This is just showing how they do it, that they put blood samples on there, they put different antibodies, whether antibody B, antibody A, and they just look for clumping. It happens like instantaneous. We don't have to wait, we don't have to grow anything up, we don't have to do anything. It happens instantaneous. So this is showing another plate that they did it in, where it clumped in B and it clumped in the Rh factor, but we don't see any of those nice big red brick clumps in the A. What's that person's blood type? B positive, which I wish I had that blood type just to be like, I'm B positive. But I don't. It'd be awesome. You want B positive? I would love to walk around and be like, be positive. O negative. Now, titrations is used to identify if a patient, and I don't even want to say a patient, because you guys might have to have this done. First, has anyone known if they've had a titer done? Some of you will. Titrations are usually used to determine if someone, so I don't even have to say a patient, because you're not always a patient, to know if someone has antibodies, and if so, at what level. Just because you have a few antibodies in your body doesn't always mean it's enough antibodies to make you immune to something. Meaning if you were exposed to something, you might not have enough antibodies to really react with it, that you may still get sick. And so, remind me again, who of you are going into nursing? Are you going into respiratory? Dental. Now remind me again, who of you has had the chicken pox? We'll have a titer done. Reason for it, and I don't think they'll do it. I don't think they do titrations for people that have had the chicken pox vaccine. But they want to know, for people that go into a healthcare field, they want to know how immune are you to things? Because if you're not immune to something, they'll make you get the vaccine. They'll make you get a booster. Because if you've had the chickenpox, because I've had the chickenpox, is there going to be a medical record on my file that I've had chickenpox? No, we didn't go to the doctor. All they're going to say is, she's got chickenpox. Go calamine lotion on it and, don't itch. It'll go be fine. You didn't go to the doctor for chickenpox unless, you generally were older when you got it and had more severe complications. And if you don't go to the doctor for something, there's no medical record that you've actually had it. So to say if I went in right now and say, yeah, I've had chickenpox, and they're like, prove it. Because if I've never had chickenpox and there's no record I had a vaccine, and I go work with a patient that has chickenpox, that could be deadly. It is definitely way worse to get chickenpox as an adult than versus a younger kid. And so they want to check anyone that goes into the medical field, where are they, on immune system level. So people that have had chickenpox, and there's no record that you've had chickenpox, and there's no record that you have a vaccine, they're going to do a titer. And what they do is they take a blood sample and they are going to dilute it out. It's called the serial dilution. So they're going to test multiple dilution factors of that blood sample with the antigen. And they really want to know, how dilute can we make that blood plasma? How dilute can we make it that there's still enough antibodies that are going to clump with antigens present? So this is showing that serial dilution. If we just take straight blood plasma and mix it with antigens, yeah, it's going to clump. There's enough antibodies there. But what if I start diluting it out, a 1 to 10 sample? Still enough antibodies present. 1 to 100, still enough antibodies present to form that clumping. 1 to 1,000, still enough antibodies to form clumping. But by the time you dilute it 1 to 10,000, there's not enough antibodies to clump with any type of antigen present. And so they say the most dilute that your urine can be, or not your urine, but your blood sample can be, that can still show a positive reaction. You can still see that clumping happening. It's considered your titer level. And based on that, if you can dilute your blood 1 to 1,000 and there's still enough antibodies present to be able to react to an antigen, you've got a strong immune system response. And they'll be like, yes, they truly have had chickenpox and they still have a good, strong immune system. We can send them to work with patients that have chickenpox. If, however, it maybe clumps here and then nothing after that, they might be like, your titer was pretty low. You probably need a booster or you need a vaccine. They do this sometimes for tetanus, but honestly, the cost of a titer, just easy. We're like, we'll just give you a booster. Like, why even run this? But they will do this for chickenpox. So it allows us to see where are the number of antibodies. Like, how many antibodies does someone have that allows them to be immune? This type of test is called the neutralization test. And really we want to see, does a patient have antibodies against some type of virus or against some type of toxin? So we're looking for the presence of antibodies in that patient sample. The patient has the antibodies. If they are there, they were able to neutralize the virus so that it can't get in and hijack and cause disease. Or if it's against a toxin, that toxin can't cause any type of disease or damage. We're just looking for the presence of antibodies in a patient. So how does this work? I've got a picture. So this is showing an example. Those are some toxins, and if I mix it with a cell, the toxins are going to kill the cell. It's the basic. Toxins are going to kill a cell. However, if we mix those toxins with a patient sample, and that patient has the antibodies, those patient antibodies are going to bind to the toxins, and so then when I then mix some cells in with it, and I look at it the next day, all the cells are still alive because the patient had antibodies that neutralized that toxin, so the toxin couldn't kill the cell. Or the same basic idea, if I mixed some cells with the patient antibodies and I mixed it with a virus, as long as the patient had the antibodies, those antibodies are going to bind to the virus and all of the cells are still going to be alive the next day. So we're looking for patient antibodies. Do they have the antibodies present against certain viruses or against certain toxins? If they do, the cells will live. Now, it's like nice, pretty pictures. In reality, you're looking underneath a microscope at cells living. This is just to show you over a time period. This is what cells, they're all smushed on a plate, look like underneath the microscope. And over the next 24 hours, they start to die. If the cells start to die over that 24 hours, does the patient have antibodies? Nope. They were not able to neutralize the virus or the toxin or whatever else they added to it. So it just helps identify, those antibodies present against certain things. We can look to see if patient has something. We can use these fluorescent dyes, and we can test for antigens and for antibodies. So I'm going to draw stuff. You know that antibodies have two arms, and they've got their little hands up here that can attach to things, whether it's little triangles or whatever it is. Antibody, we know it can attach to things. And we really want to look to see, are antibodies there? Are they attaching to something? So one thing we can do is attach something else to the antibody. We can attach things to that body part of the antibody. We can make this some little fluorescent dye. And so if the antibody is present and it's sticking to something, underneath a microscope, we have microscopes that will fluoresce. They have a UV light. If we had antibodies against this particular bacteria, and we know it's a very specific bacteria, because if you looked under a microscope, you're like, oh, no, they look round. There's so many bacteria that are like a cocci-shaped bacteria. But if we had the correct antibody, we're like, well, I'm going to add the antibody against Streptococcus pyogenes. And if I looked underneath the microscope and I see everything just glowing like crazy, I know that that antibody stuck to that particular bacteria, and it will glow, and it's super easy to detect. Now, you can also use it to detect antibodies as well. Can an antibody stick to an antibody? It can. Antibodies can stick to antibodies. So the idea is that normally they stick to antigens, but those little on the antibodies themselves, they can stick to another antibody. They stick to the body portion of it. And so if an antibody is present in a patient sample, I can have very specific antibodies that recognize antibodies. They're called antibodies. It's horrible. But antibodies can recognize other antibodies. And if you see things fluorescing, you know that they found whatever it is that they were supposed to, and they now glow underneath the microscope. One way that we kind of take this idea, but without the glowing, is something known as an ELISA test. It's a labeled antibody test. So we label it, I'm going to use a different color. We still stick some type of enzyme on the antibody. Jobs are to take substrates, do something with them to make a product. That's the job of an enzyme. They're not used up. They just take something and make something else with it. So it's called an enzyme linked because I'm going to add an enzyme to an antibody. So I'm not going to be looking for any kind of fluorescing. But eventually I'll add a substrate. And if the enzyme is present because the antibody is there, I will see a color change. Now, I'm going to see if this link will work, if I've got audio in here. We can use the ELISA test to detect both for antigens and antibodies. It's extremely sensitive. So truly, if you have one antigen in your body, you're going to see a color change, or in your sample. If you just have one antibody in that sample, you will see a color change. And so it's extremely sensitive. It's easy to perform because we have machines that do it all for us. And you can test almost 100 samples all at the exact same time. So you can test a lot of samples, easy to do, super sensitive, So it's done all the time. Now, how they do it, yeah, I'll leave it up here. See if I can do it. They have these little plates. They're plastic plates with little tiny divots or wells in them. So here's my little well. And again, there's two different ways to test for it. There's what's known as a direct ELISA. A direct ELISA, just like a direct immune system test, is testing for the presence of an antigen. On this plate, we are going to put antibodies. What do we want to test for today? What's a nice bacterial disease? Throat. All right. We're going to test for strep throat. We are going to add antibodies that are specific for strep. Then we're going to add a patient sample. So we have antibodies that are going to stick to these wells. These wells are made so that things can stick to them. So we're going to add it. They're going to stick. Then we're going to add a patient sample. And we're going to assume our patient has, a different color, strep throat. That means they have the bacteria. I'm using a big thing. Just looks like. This is the strep bacteria, because that's from the patient. It's now stuck to that antibody. That's not going to give us a color change yet. What they're going to add next is that enzyme-linked antibody. So they're now going to add another antibody that is specific for strep, but this one has that little enzyme tag on it. Then they're going to add a substrate. And if this antibody is there, and it's only there because the patient had strep, if the patient didn't have strep, it wouldn't have anything to stick to. Because that enzyme is now there and they add a substrate, that substrate will start to turn colors, and you will see colors show up in the plate. So this plate can to test for 96 patients all at once. So we can test 96 different patients for strep throat. Can you visually see who has strep throat? Yeah. Now, are some of the blues darker than the others? Yeah, which usually means there is more antigen present. So it also somewhat helps us quantify it a little bit. We don't always say for sure that we use it for quantifying because it could have been a bad sample collection. But whether it's some of just the palest blues, it doesn't matter. Any blue indicates that that particular antigen is present for us, strep throat, and the patient can now be tested and given antibodies for it. So this is the direct test. We are testing for that strep throat. But we can do this as well as an indirect test. Test for antibodies. We can test to see if maybe a patient's recently had strep throat. Same colors. As we can add the strep throat antigen to the plate. I get 3 of them. Positive reactions, just like I can do a positive test up there. But we have strep antigens we can add to the plate. Then we're going to add the patient sample. If the patient has antibodies for strep throat, it will recognize it. So the patient antibodies are going to recognize strep throat, and they're going to attach to it. Good job. We still don't have that enzyme linked yet. So now we're going to add our enzyme linked antibodies. And we can add enzyme linked antibodies that can recognize antibodies. And it will recognize this body portion. Now the only way that these enzyme linked antibodies will stick is because the patient had antibodies which eventually stuck to those antigens. Then I add a substrate, and because that enzyme is there in the plate, stuck to this big huge stack, you'll get a color change. And it will, again, tell us really fast, 96 different patients we can test all at once. The machine does this all for us these days. And we will immediately be able to see who has antibodies for strep throat or whatever else it is that we're testing. If I can get this, because this is a nice visual explanation of just that. I feel like there's still some places that can open it. I really do hate technology. Well, I'm not going to spend any more time looking it up. Bummer. I'll see if I can find it and I'll post it on there. It's a nice summary of, actually watching them like add stuff and like rinse stuff out and then the color change happening. But it's done quite often just because, again, a machine can do it all for us, and you can test 96 different people for one particular antigen or antibody, or you can test one patient for 96 different things. So it's used quite often. Now, the Western blot test, the same idea for an ELISA is that we're still going to be using an enzyme-linked antibody. But a Western blot is done a little bit different because we're not detecting just the presence of one single antigen. We're detecting multiple antigens, and we're detecting multiple antigens based on size. So we're looking for multiple antigens, and we're looking at the different sizes of those antigens, knowing that each antigen is a different size. Some antigens are really big, some are really small, and we're looking for the presence of as many of them as possible. So what they'll do is they'll take a patient sample and they'll put it in a gel, kind of just like that auger we have upstairs, except it's square now. They'll put it in a little tiny well. They will add some electricity to it, and because things will move toward the negative part of the electricity, and they'll do it based on size. The smaller the antigen, the easier it is to kind of wiggle through that gel, that agar material. The smaller the antigen, the farther it will travel over time. The bigger antigens kind of get stuck in place. They can't travel very far. And so it separates out all the antigens just based on size. So that's first all we do is we just separate all the antigens that are present based on size. Then we're going to add an enzyme-linked antibody to detect all of those different antigens so that we can visually see them. Now, it's called a Western blot for a reason, because we're going to take a gel, just like that auger upstairs, but we need to get those antigens on a paper. And so the easiest way to do it is, they say absorbent paper, a sponge, is we take a wet sponge, we take that auger, that gel, we take the paper that we want it to go on, and we take it's a filter paper, big old stack of paper towel. And if you take paper towel and you push it down on a sponge, what's going to happen to that paper towel? It's going to suck all the water up, which is the idea. Is if we take dry paper towel and we put it on top of a sponge, it's going to suck all that water from the sponge. As it does that, as the water goes from the sponge up into the paper towel, any of the antigens that are on this gel are going to travel with the water, and they're going to deposit themselves on the paper. It's kind of cool. It's like, I know this looks all whatever, but in reality, it's literally paper towel you're squishing on a sponge in a lab, and then all the antigens on your gel just go up with the water. They just get whipped up with the water and deposit themselves onto paper. On the paper, you add those enzyme linked, and we can visually see now, kind of just like those test strips upstairs, you can visually see the presence of all the different antigens and based on size. Because there are certain diseases that it's not looking at just the presence of one antigen to say that a patient has a particular disease. Because a patient may have a few antigens for a particular disease, but they don't have them all because their body could have broken them down and only some of them are present still in the body. We need to look to say, no, they truly have all the antigens, which means they truly have that particular organism in a large quantity in the body. Now, the only disease that I know that Western blot is routinely used for, and it's used for quite a bit for patients in the Midwest, is Lyme's disease. So Lyme's disease, which I think is my next slide, the CDC says for someone to have Lyme's disease, they have to show that they have at least 5 of the 10 Lyme's disease, the bacterial antigens present. If you just have 1 or 2, you'd be like, you probably don't actually have it. Your body's probably, may have been exposed to it, but it's not in enough present to say you actually are suffering from that particular disease. And since Lyme's disease is pretty common around here, the Western blot is done for Lyme's disease. So this is showing the number 1 column is just a negative control to make sure nothing's contaminated. The number 8 column is a positive control showing lots of possible antigens. And then you have patients, 2, 3, 4, 5, 6, and 7. Who has Lyme's disease? 3. And I would say 2 as well. Yep. So patients 2 and 3 do. Patients 4, 5, and 6 and even 7 are all showing that they've got some antigens present, but again, not enough to say that that person has an active infection. Some of that may be at some point you were exposed to something, antigen, something similar to that antigen is still in the body, but it's not an active infection. Not enough that you would be showing signs of this particular disease. So if I go back to this one, we have patients 1 through 6. Who has Lyme's disease? 3. 5. I would even say 6. Yep, I was going to say they are not dark, but they are present. And as long as you've got any color change whatsoever, even if it's a faint blue line, that means that that antigen is there. So if you know anyone that's ever tested positive for Lyme's disease, whether it was people, whether it was pets, they do Western blot tests. They do take a lot. I don't even think the local hospitals do the Western blots. I think the last time I checked, they ship all the samples out to like a lab in California that just does Western blots all day long. So sometimes samples can take a little bit of time to get results back. And the last group of immune system testing that we have are what are known as point of care testing, which means they're super simple immunoassays that give us results in minutes. Meaning we are looking for that antigen-antibody combination that's going to give us a nice little color change, and it's going to tell us something in minutes. Because if you go to the doctor and you're not feeling well, you want to know as fast as possible what's going on. You really don't want to be told, we're not really sure. Can you come back in a couple days? Because you're going to be really mad because you probably just paid $200 for that. You want quick results. You want to know what's going on because you want quick treatments. So a couple different ones. One is known as an immunofiltration assay. Is they will just add 1 drop of patient sample right onto this tiny little disc, and that little disc has an antibody already labeled on it. And if the patient sample has the antigen, that little circle will turn a color. It's called it's immunofiltration. It's a little filtration paper. One that might be more common is known as an immunochromatography assay. Same kind of idea is that it still is a type of filter paper. It still has antibodies labeled on it. And it's still going to be looking for antigens, except those antigens have to travel up a test strip. An example of an immunochromatography assay is a pregnancy test. It would also be the test they use for mono. It would also be the test we use for COVID, any of those at-home COVID tests. All use the immunochromatography assay. Also, what did we just test for upstairs that uses the same thing? the strep throat. So these are all immunochromatography assays. We add a sample into one particular area, we wait for it to travel through something, and then we're looking for that particular antigen-antibody reaction. That antibody that's labeled on there has some type of color reaction enzyme or substrate on it that when it reacts with the antigen, it will show up as a color. And again, the reason why they always have one line is to ensure that you had enough of a sample to get to that point. So one line will always mean negative on any particular sample. That one line just means you did it right. It's that second line that you're looking for. So that's why I'm like, this is an immunochromatography dipstick. That's what it looks like, is we added some of the antigen possibly from your samples, and we are looking to see if it mixes with some of the antibodies that are up there, and it will give us a color change. Before I see you next Monday, since we did finish this PowerPoint, before I see you Monday, so Sunday night, there is a quiz on this. Other thing, they're not due before I see you Tuesday, but they're coming up really soon. There is the exam on the immune system due next Tuesday night. So if you're like, oh my goodness, my Tuesday, not Tuesday, Monday. Again, I keep thinking this class is Tuesday, Thursday. It's due next Monday night. So if you know you've got a really busy next Monday, you may want to get that immune system test done and out of the way before we get to Monday. But the immune system test is due Monday. If you haven't done that chapter 16, that's the immune disorders, it's due, I think it's due next Friday, but it's 10 points. Again, looking at some of those autoimmune disorders, allergic reaction type of things, that's an immune disorder. There's a 10-point quiz that goes along with that. Then we can be done with the immune system. On Monday, we're going to start our next little mini unit, all on genetics, so looking at DNA and transcription translation. I think it gets 2 lectures, so it's like a little mini. Otherwise, have an amazing